Chapter 3 is titled The Molecules of Cells. When we say that a molecule is organic, that means that it has both carbon and hydrogen. If we are simply referring to hydrocarbons, hydrocarbons are organic molecules that just have carbon and hydrogen. Now, hydrocarbons, those are the simplest organic molecules, but organic molecules can have other elements other than carbon and hydrogen. This picture right here is just showing how hydrocarbons can come in a variety of shapes. You don't need to memorize any of these names. Hydrocarbons are all nonpolar compounds. If you remember from chapter 2, we said for a compound or a molecule to be polar, there has to be an unequal sharing of electrons, which means there would have to be a electronegative element like oxygen or nitrogen. And hydrocarbons, we don't see any of those here. Nonpolar compounds, they are always hydrophobic, which means that they are not water soluble. So phobia meaning fear, water hating. We also said in chapter 2 that like dissolves like. Since water is polar, it can only dissolve other polar compounds. What we can also see in this image is some isomers. Isomers are compounds that have the same molecular formula but different structures. If I counted the amount of carbons and hydrogens in 1-butene and 2-butene, they're the same. But you look at them and structurally, the way that these atoms are put together, it's different. So isomers have different properties. They behave differently in chemical reactions, and this is because the structure of a molecule dictates its function. If two molecules, even if they have the same formula but they have a different structure, they will have a different function. Functional groups are these add-ons that I can put on hydrocarbon skeletons. If I look at most of these functional groups, so all of these right here, they are all polar except my methyl group. The reason why these ones are all considered polar is because they have an electronegative oxygen or nitrogen that's going to hog some electrons, which means if I put one of the polar functional groups on a molecule, I'm going to make it hydrophilic. It's going to be able to dissolve in water because like dissolves like. Water is polar, so it will dissolve other polar molecules. If I take a look at these functional groups, a hydroxyl group is an OH. If I add an OH onto a molecule, I can call that molecule an alcohol. Carbonyl group is a C double bonded to an O. If that carbonyl group is at the end of a molecule, I can call it an aldehyde. If the carbonyl group is in the middle of a molecule, I can call it a ketone. A carboxyl group is if I look at the structure of it, it's a C double bonded to an O and an OH. So it's really a carbonyl and a hydroxyl put together. One thing I want to point out about carboxyl groups is they can make molecules act as acids. Reason is because you see right here, this molecule is going to donate an H. I can see right here that when it's ionized, it's missing an H, so it has donated H pluses to the solution, so it's acting as an acid. The amino group is a nitrogen and 2 hydrogens. Amino groups can actually make molecules act as a base. Remember, bases, they are H plus acceptors. So I can see first this amine, which is a molecule with an amino group, does not have an H. And then here, it has accepted an H. We've got phosphate groups. Phosphate groups, I can see the structure here. It's 1 phosphorus to 4 oxygens. We'll look at phosphate groups with ATP, and then a methyl group is 1 carbon bonded to 3 hydrogens. This is a compound that has a methyl group added to it. We'll see how methyl groups are important when looking at DNA and how to express certain genes. The 4 big organic molecules we are going to be studying are carbohydrates, lipids, proteins, and nucleic acids. Because they're large organic molecules, we can call them macromolecules. Sometimes they're called biomolecules because they make up cells. We are only going to focus on carbs, lipids, and proteins in this unit because nucleic acids, that's DNA and RNA, we're going to dedicate a whole unit to that. So all of these larger molecules, we can call them polymers. And all of these polymers are made of many monomers. Poly means many, mono means one. So for an example, this one circle would be representing one subunit, so I could call this a monomer. And then once I start putting multiple monomers together, then this is going to make up a large polymer. The terms polymer and monomer, they are not specific to carbs, lipids, or proteins, or even nucleic acids. I can just use these terms to apply them to specific organic molecules. If I want to link monomers together, the process that accomplishes this is called dehydration synthesis, or we could call it a dehydration reaction. This is when we remove water to form a covalent bond. So if I look at this picture, again, I'm not using actual structures. I'm actually just using these purple circles to represent one circle is a monomer, a bunch of them together is a polymer. What you will always have on one end, an OH or a hydroxyl, and then on another end you'll have an H. So what'll happen, dehydrate, we're going to remove water, these will combine, water will leave, and then we have created a new covalent bond. If we want to separate monomers, this process is called hydrolysis. So hydro, it can mean hydrogen, it can also mean water. Lysis means to split. So this is the opposite of a dehydration reaction. With hydrolysis, I'm actually adding water to break a covalent bond. So again, hydro, water, lysis to split. If you look at this picture, it's the opposite of what's happening with dehydration synthesis. Water is now being added. The water will be split so that you have an OH on one end of a monomer and an H on the other, and now you have broken this bond. So now if we start getting into the specific types of macromolecules, we'll start with carbohydrates. The monomer of a carbohydrate is a monosaccharide. A monosaccharide always has a formula that has a multiple of CH2O. So for example, here is a type of monosaccharide, and its formula is C6H12O6. It's a multiple of CH2O. Other terminology, if I ever have a monosaccharide or a disaccharide, where a disaccharide is linking two monosaccharides, I can refer to these as sugars, and they typically taste sweet. These are what I would call a simple carbohydrate. My complex carbohydrates, these would be polysaccharides, where poly means many, and I'm going to put a bunch of monosaccharides together. An example of a polysaccharide is starch, and these typically don't taste sweet, and we can refer to those as our complex carbohydrates. We've already defined isomers, so now I'm going to point out an example of isomers of monosaccharides. There are different types of monosaccharides. Two types are glucose and fructose. Now I underlined the ose in both of them because typically if you see an ose, that's going to be your clue that this molecule is a carbohydrate. Glucose and fructose are isomers because they both have the same chemical formula of C6H12O6, but if you look at the structures of them, they are different. Glucose has a carbonyl group at the end, fructose has a carbonyl group in the middle, and then because of that, the locations of some of these hydroxyl groups are different. It's important to note then that because these are isomers, they have different structure, they also have different functions. They taste differently. Glucose is the main fuel molecule of cells. So here you see glucose in the straight chain form. Glucose also does come in this cyclical form. This picture is an example of dehydration synthesis where two monosaccharides are being linked together. So if I label this arrow right here, this is the process of dehydration, synthesis, where if you remember what that means, dehydrate means you're taking out water. So this H and OH, they are going to be removed to form water, and now I have linked these two monosaccharides together. I formed a new covalent bond. So glucose and glucose, if I put two of those together, I make the disaccharide. Di means two sugar molecules, maltose. This is the sugar that's in malted milkshakes. And now let's say I eat maltose sugar and my body wants to break it down. The opposite then, if the arrow goes the other way, that's going to be hydrolysis. Where hydrolysis, I am adding water in order to break the bond between monomers. So a monosaccharide is basically one sugar molecule. It is the monomer of carbohydrates. If I put two together, I get a disaccharide. Now, if I put many together, I get a polysaccharide. So there's three main types of polysaccharides we're going to focus on. They are starch, glycogen, and cellulose. If I look at what they all have in common, they all are a bunch of glucose monomers put together. What is different about them is the way the glucose molecules are structured, all right? I can see that each of them has a different shape to them. And since structure dictates function, they are going to all have different functions. So let's take a look at starch. If you look at the structure of starch, the glucose molecules are kind of in this helical form. Starch is how plants store excess glucose. So whenever you are eating a starchy food, it had to have come from some type of plant material. Second type of polysaccharide, glycogen. If we look at the structure of glycogen, it is more branching. Now, glycogen is how animals store excess glucose. We store glycogen in our liver and our muscles. So when we eat starch from plant products, what happens is our digestive system breaks it down into individual glucose molecules. We can use some of those glucose molecules for energy, but we can also store some as glycogen. Third type of polysaccharide, let's look at cellulose. So cellulose is these parallel fibers of glucose monomers, and they are linked by hydrogen bonds. So all of these dots, they are representing a hydrogen bond. The function of cellulose is it gives structure to plant cell walls. Cellulose is actually what makes up the fiber in our diet. It is the cellulose. We do not have enzymes to break down cellulose, so that's why we cannot get energy out of fiber. It really just helps keep everything in our digestive system going. Let's move on from carbohydrates now to lipids. Now, what makes a macromolecular lipid is that it is hydrophobic. It does not mix with water. What we learned about already is that anything with an electronegative atom like oxygen and nitrogen, that would make something polar and hydrophilic. So you might be thinking, well, lipids, if I look right here, if this is the structure of a lipid, I do have some oxygen. But there is an overwhelming amount of carbon-hydrogen bonds. Carbon and hydrogen being bonded together, that is a nonpolar bond. So since there's mostly nonpolar bonds in lipids, that's why they act hydrophobically. There are different types of lipids, and each type of lipid has a different function. So one type of lipid are fats and oils. Now, fats and oils collectively are called triglycerides. The reason that there is this tri is because the structure of lipids actually has two different types of monomers, one glycerol head, and then three fatty acid tails. The function of triglycerides, it's long-term energy storage. Our cells will use carbohydrates first for energy, and then they will go to lipids. Lipids are more of a dense form of energy. They actually store 2 times the amount of calories than a carbohydrate does. Lipids also cushion organs and helps insulate animals, and then they also allow for buoyancy because lipids are less dense than water, so that helps with aquatic animals. Let's take a look at dehydration synthesis happening to make a triglyceride. So here we have a glycerol molecule, and actually the OL comes from the fact that glycerol has these hydroxyl groups, which makes something an alcohol. That's where the OL comes from. And then here's a unlinked fatty acid. It's an acid because over here it has a carboxyl group, which can act as an acid. So dehydration synthesis, I remove an H2O, and now I've made this new covalent bond. That would have to happen 3 times in order to make a triglyceride. There are 2 types of fats, or 2 types of triglycerides, saturated and unsaturated. Saturated fats have single bonds only in the fatty acid tail. So I'm not talking about these bonds, I'm talking about all of these single bonds. Saturated comes from the fact that they are saturated with hydrogens. Carbon always wants 4 bonds, so you can see that I have a lot of hydrogens in these saturated fatty acid tails. Because these saturated fatty acid tails are straight, this allows the molecules to stack on top of each other, so saturated fats are solid at room temperature. An example of a saturated fat would be butter or any type of animal fat. Unsaturated fats, they have at least 1 double bond in the fatty acid tail, and what that does is it puts a kink in the tail. So now anything that has one of these kinks, these molecules cannot stack on top of each other, so unsaturated fats are liquid at room temperature. An example of an unsaturated fat, that would be vegetable oil or any type of oil that comes from a plant fat. And if we go back to, well, saturated with hydrogens, not saturated with hydrogens, so if you look right here where there's that double bond, since carbon only wants 4 bonds to fill its outer electron shells, when I put a double bond, I'm actually taking away some hydrogen. So while these are saturated with hydrogens, these are unsaturated with hydrogens, not at the maximum amount of hydrogens. Another type of lipid is a phospholipid. So this is separate from triglycerides, or my fats or oils. Phospholipids make up cell membranes. If we look at the structure of a phospholipid, it is similar to a triglyceride in the fact that it does have a glycerol head and fatty acid tails. However, it only has 2 fatty acid tails. And then there's also this phosphate group and another molecule that is connected to the glycerol. Waxes are another type of lipid. Waxes, they're very hydrophobic. So waxes, they are in this straight chain form. And remember, all lipids are hydrophobic. They all have a majority of nonpolar bonds. So you can find wax on the surface of leaves, and what they do is they prevent water from evaporating out inside the leaf. Also insects, they have their outer skeleton, their exoskeleton has a waxy layer. That's what makes them shiny. And again, it's going to prevent water from evaporating from inside the body of the insect. So my last type of lipid is a steroid. What makes a lipid be considered a steroid is that it has a ring structure to it. Now, there are different examples of steroids. Cholesterol is an example of a steroid. It has a ring structure. Cholesterol has 2 main functions. One function is that it makes up part of animal cell membranes. It also helps to serve as the starting point to make other steroids, such as sex hormones. So another type of steroid are your sex hormones like testosterone and estrogen. So you can see that both of these have a ring structure to them. That's why they are a steroid. And what actually makes testosterone and a type of estrogen, estradiol, different is that we can see that there's different functional groups here. Moving on from lipids, let's talk about the last macromolecule, and that is proteins. The monomer of proteins is amino acids, so meaning you put together a bunch of amino acids to make a larger polymer of a protein. There are actually 20 different amino acids, so you can see here this lists all 20 of the different amino acids. Because there are 20 different amino acids that can come together to make a protein, there's a huge diversity of what proteins can do. So some proteins are enzymes, which help speed up chemical reactions. Other proteins serve a structural purpose. They make up hair, tendons, ligaments. Other proteins can contract and they help organisms and cells move by making up their muscles. Some proteins are antibodies, which aid in the immune system. Other proteins are hormones, which are chemical messengers. Now, most hormones are protein hormones, but there are some lipid steroid hormones that I just mentioned, like estrogen and testosterone. There are also some proteins that have a transport function. So for example, hemoglobin, that is a protein in our red blood cells that transports oxygen. You also have proteins embedded in cell membranes in order to help move things in and out of cells. If we look at the structure of an amino acid, which is the monomer of a protein, all 20 amino acids have the same basic structure where first you have a central carbon. Sometimes that central carbon is called the alpha carbon. And then coming off of that central carbon, there is an amino group, which is why they're called amino acids. And then there's also a carboxyl group. And remember, I told you that carboxyl groups can donate this H and act as an acid, so that's where the name amino acid comes from. Also coming off of this central carbon is a side chain or an R group. The reason R is used is because there's nothing on the periodic table that's just an R. So this is actually what's shown in blue here. This is what changes between all 20 amino acids. And then since carbon always does want 4 bonds in order to fill its outer electron shell, in order to give carbon the 4th bond, that's why there's this hydrogen. The R group determines the property of the amino acid. A lot of times I'll write AA for amino acid. So if I look at one amino acid, leucine, I can tell that this is the R group because here I have the central carbon, the amino group, the carboxyl group, and then the hydrogen. So this is my R group. You should be able to look at this R group and tell me that this R group makes the amino acid hydrophobic, and that's because it has all carbons and hydrogens, which form nonpolar bonds. Nonpolar, hydrophobic, they are synonyms basically. Another amino acid, serine. So you should be able to tell me that this is the R group because I can identify the central carbon, the amino group, the carboxyl group, and then the hydrogen. Serine is hydrophilic because it has this electronegative oxygen that's going to hog electrons from the hydrogen that it's bonded to. If I want to link amino acids together, again, it's a dehydration reaction or dehydration synthesis. So if I look here, I have 2 amino acids that I want to link together. These amino acids, they're not showing the specific R groups, they just have the R. But what's going to happen is an OH from the carboxyl group is going to join together with an H from the amino group. These two are going to form my water, and now I'm going to have, where this space was, a new bond form. Now, a peptide bond is what we call the covalent bonds that link amino acids together. Whenever you have peptide, it has to do with proteins. So other common terms, this would be a dipeptide because it is 2 amino acids linked together. Once I start putting more amino acids together, I will call that a polypeptide. Poly means many. Now I have multiple amino acids put together. Of all of the macromolecules, proteins definitely have the most complicated structure. There's 4 levels of structure, and we're going to go through those right now. primary structure, it is the simplest level of protein structure. So this is first just the order of amino acids put together, and it is the specific order of amino acids. So like over here, glycine, threonine, glycine, glutamate. This primary structure is actually what's coded for by the DNA. We'll learn how that happens in Chapter 10. Secondary structure now, that amino acid chain, which was the primary structure, is going to either fold or coil. The coiling of a secondary structure is called an alpha helix. The folding of the secondary structure is called a pleated sheet or a beta pleated sheet. Primary structure, that's held together by those peptide bonds between amino acids. Secondary structure is actually held together by hydrogen bonds. If I look at where the hydrogen bonds are connecting, it's being connected by a hydrogen from the amino group attracted to an oxygen from the carboxyl group. So secondary structure, it's just this local folding that has nothing to do with the R groups. The tertiary structure, this is really where proteins get their 3D shape. Some tertiary structures are more of a globular shape, where some are more long and fibrous. How tertiary structure forms is by interactions of the R groups. Remember, R groups have different properties. So if you have some R groups that are hydrophobic and some that are hydrophilic, the hydrophobic ones that are afraid of water or don't mix with water, they're going to hide in the middle of the protein. The hydrophilic ones, they are going to go to the outside of the protein. If I have R groups that are polar, I can have hydrogen bonds form between them. I can have ionic interactions between some R groups if one R group is positively charged and one is negatively charged. There is also another type of interaction of R groups called a disulfide bond, where the amino acid, so cysteine, or the abbreviation is CYS, as is a type of amino acid, it has sulfur in it. So these disulfide bonds can form between two cysteines. And this is really just a type of covalent bond. So you can see why the primary structure that has the order of amino acids, that really does shape the tertiary structure, because where those amino acids are with different R groups, that's going to dictate how this 3D structure starts coming together. All proteins have primary, secondary, and tertiary structure. Some proteins have a fourth level of quaternary structure. This is when proteins are made of different subunits. I can also say that they are joined polypeptide chains. You can also think that quaternary structure is basically proteins made of multiple tertiary structures put together. The subunits of quaternary structure could be identical or they could be different. So I could have two of basically the same proteins held together or two different ones held together. The way they're held together, it's going to be the same interactions that hold together tertiary structure. Hemoglobin is an example of a protein that has quaternary structure, where it actually does have 4 different subunits to it. 2 of them are the same, and that's why these are both shown in orange. And then 2 of them are different, these ones shown in pink. You're going to see that we're stressing a lot that structure dictates function, and this is especially important for proteins that have such a complicated structure. There's actually a special name for when proteins lose their shape. We say that that protein has been denatured. What can cause proteins to lose their shape is things like changes in salinity, changes in pH, extreme heat. Most proteins have an ideal environment where there's an ideal salinity, an ideal pH that they work best in. Same thing with temperature, an ideal temperature. If you've ever seen an egg being cooked, which I'm sure most of you have, you see that the egg white, which is protein, it is turning from clear to white. What you are witnessing is the extreme heat from the stove denaturing those proteins. So far, we've learned about 3 types of macromolecules that all cells are made out of: carbohydrates, lipids, and proteins. The 4th one is nucleic acids. Now, this is just going to be an intro to nucleic acids because we will learn about them more in detail at a later unit. But there are 2 types. There's DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. DNA is the genetic material inherited from parents. A gene is a segment of DNA that codes for the amino acid sequence of a protein. So really, DNA codes for our traits by coding for all of the different proteins that we are made out of. And we already saw that there's a huge diversity in protein function just because there's 20 different amino acids, and proteins are made of hundreds to thousands of different types of amino acids. Now, one of the important things that allows DNA to be this genetic material that passes information from generation to generation is that the DNA that is in a cell can make a copy of itself before that cell divides to make sure that no genetic information is being lost. So if DNA codes for proteins, then what does RNA do? Well, RNA is the other type of nucleic acid that DNA does need in order to make proteins. So we're going to learn more in detail again at a later unit that DNA is first transcribed or rewritten into the other type of nucleic acid, RNA. And then it's actually RNA that is read or translated to make the protein. And remember, first, the primary structure of the protein is just the order of amino acids that's coded for by the DNA, but then there's going to be that other folding process that takes place. The monomer of nucleic acids is a nucleotide, and this is true for both DNA and RNA. Nucleotides have 3 different parts. There is a phosphate group, which is one of the functional groups. There is a 5-carbon sugar, and then a nitrogenous base. An example of nitrogenous base is adenine. It's called a nitrogenous base because you can see that there is nitrogen in it. Here I'm going to highlight, though, what makes these nucleotides different in DNA and RNA. So I can see that both of them have a phosphate. Both have a sugar, but in DNA it is deoxyribose sugar. The molecular structure is a little bit different, where I have ribose sugar in RNA. And then each of them does have 4 nitrogen bases. 3 of them are the same. So both of them have AGC, which stands for adenine, guanine, and cytosine. But then the 4th base, DNA has a T base, or thymine, and RNA has a U base, or uracil. Just like with the other macromolecules, a polymer is built from all of the nucleotide monomers by a dehydration reaction. The polymer I can also call a polynucleotide. Here you can see how they're coming together, where here I have a nucleotide with the phosphate, the sugar, and the nitrogenous base. So this nucleotide is going to be connected to the other one by the sugar and the phosphate. So you'll see what happens is that the sugar and phosphate, they form what we call the backbone of nucleic acids, and then the bases are facing inward. Now, typically, the polymers do come together differently in DNA versus RNA, where DNA is mostly double-stranded, or we could call it a double helix, and RNA is mostly single-stranded. Here's a closer look at how the nucleotides come together to form this double-stranded double helix in DNA. So you can see I still have the sugar-phosphate backbone on either side, and then I have the nitrogenous bases coming together in the middle. There's always going to be C and G paired together and A and T paired together. I'm always going to have those base pairings. And then there's another type of bond, which we've discussed, that pair together the nitrogenous bases, and these are the hydrogen bonds.